A stimulating experience. Cortical electrical stimulation mapping is a procedure that was developed in the mid-20th century by a neurosurgeon named Wilder Penfield that used a small electrode to stimulate an exposed brain in specific locations. During this procedure, the different responses were recorded. This process could allow neurosurgeons to take out damaged brain tissue and preserve healthy brain tissue. This process also opened the door to discovering more information about the human brain. Specialized cells make up the nervous system. A neuron is a nerve cell, and it is comprised of a cell body, dendrites, and an axon. A glial cell is a non-neuronal brain cell that functions as support through structure and nutrition. The neuron doctrine is a hypothesis that the brain is comprised of distinct cells that differ structurally, metabolically, and functionally. A synapse is a small gap where information is exchanged between neurons. A mitochondrion is an organelle found in the cell that is responsible for metabolic energy in cell processes. The cell nucleus is a spherical central structure of a cell that contains genetic material. Ribosomes are structures in the cell body where genetic information is translated to produce proteins. All neurons contain certain features. These features are an input zone, an integration zone, a conduction zone, and an output zone. The input zone is where information is received from neurons or other types of cells. The integration zone is where information is integrated and the decision to send a neural signal is determined. The conduction zone is where information is transported, and the output zone is where information is conveyed to other cells. A dendrite is an extension of the neuron that receives external signals. This is regarded as the input zone. The cell body, or soma, is an area identified by the presence of the cell nucleus. This is regarded as the integration zone. The axon is a singular extension from the neuron that carries action potentials from the cell body to other cells. This is regarded as the conduction zone. An axon collateral is a branch of an axon from a single neuron. An axon terminal is also known as a synaptic bouton. This is the end of an axon or axon collateral that forms a synapse on a target cell. This is regarded as the output zone. There are three main types of classifications for neurons. These are multipolar, bipolar, or unipolar neurons. Multipolar neurons have many dendrites and a single axon. This is the most common type of neuron. Bipolar neurons have a single dendrite and single axon on opposite ends. This is most common in sensory systems. Unipolar neurons have a single extension regarded as an axon that splits into two directions, one direction serving as the input zone and the other direction as the output zone. These neurons transmit touch information from the body to the spinal cord. In these three classifications, the dendrites are all found in the input zone. Motor neurons or motoneurons are a type of nerve cell that control muscle and gland movement. Sensory neurons are a type of nerve cell that is influenced by the environment. An interneuron is a type of nerve cell that receives signals and sends out information. An interneuron is neither a sensory neuron nor a motor neuron. Arborization is dendrite branching. Arborization illustrates the complexity of the information processing that a neuron is responsible for. A synapse is usually comprised of three main elements: a presynaptic membrane, a postsynaptic membrane, and a synaptic cleft. The presynaptic membrane is the membrane that sends signals out by releasing neurotransmitter. The postsynaptic membrane is the membrane that accepts information and responds to neurotransmitter. The synaptic cleft is the space between presynaptic and postsynaptic membranes. The synaptic vesicle is a circular pouch that holds a neurotransmitter. The vesicle fuses with the presynaptic membrane and ruptures. Neurotransmitter is the chemical that is released by the presynaptic axon terminal. Neurotransmitter allows communication between neurons. Neurotransmitter also diffuses across the synaptic cleft and interacts with receptors. A receptor is a protein that binds and reacts to molecules of a neurotransmitter or hormone. Neuroplasticity is the ability of the nervous system to change in response to environment or experience. The axon hillock is a cone-shaped projection from the cell body. This is the specific area known as the integration zone. Neurons almost always have one axon, and this axon splits into several axon collaterals. Innervate means to provide neural input. A motor protein is a kinetic protein molecule that transports cargo from one location to another within a cell. Axonal transport is the transportation of substances from the neuronal cell body to far regions in the dendrites and axons, and from the axon terminals back to the cell body. This movement of material moves in two directions. Anterograde transport moves material toward axon terminals, and retrograde transport moves used material back to the cell body to be recycled and used again. Fast transport occurs outside the axon. Slow transport occurs inside of the axon. Visualizing the cells of the brain. Histology is the scientific study of the composition of tissues. This study experienced a revolution in the mid-1800s when someone discovered that fabric dyes could stain cells and detect previously hidden structures. A Nissl stain is a type of cell stain that stains RNA and reveals all cell body, used to measure the size and density of cells. Golgi stain is a type of cell stain that stains a small portion of neurons darkly and completely. This stain is used to identify shape and the amount of neurons in a cell. Autoradiography is a histological technique that shows the distribution of radioactive chemicals in tissues. Immunohistochemistry, or IHC, is a technique in which labeled antibodies are created and used to visualize histological distribution of target proteins. In situ hybridization is a technique in which labeled complementary nucleic probes are used to identify cells expressing specific messenger RNA transcripts, reflecting the activation of specific genes. This method labels only neurons in which a gene of interest has been turned on. A track tracer is a compound used to identify routes and interconnections of neuronal projections.